Hi everyone. So right off the bat, if you can hear my dishwasher, sorry about that. I should have not turned that on right before I was going to record this, but oh well. So hopefully it's not too distracting. So we're going to be kind of switching gears here now for the next couple lectures. And we're going to be focusing primarily on plant anatomy. And we're going to start at the most basic and then build up complexity. So today we're going to be looking at plant cells. But first we are going to review. So last lecture we looked at angiosperm germination. And to begin with, we defined angiosperms as plants that have flowers, as opposed to gymnosperms, which are plants without flowers, such as cedars or pines. Angiosperms, by and large, with the exception of lumber, by and large in agriculture, we are cultivating angiosperms. So, they are plants with flowers, and then we said within angiosperms, we could divide them up into monocots and dicots. And monocots were plants that have one cotyledon, or one seed leaf, and those are typical of our grasses, whereas dicots have two seed leaves, or two cotyledons, and those are our trees and shrubs and some of other herbaceous species as well. Same with monocots, they're not just grasses. And then we looked at the different types of germination, or at least pattern of germination. And we started with hypogeal germination, which is typical of our monocots, and this takes place primarily underground. And we came and we started with looking at the seed, and we said the outer layer of our seed, we have our seed coat. And then in our monocots, typically we have a large store of endosperm. And this is a food store. It's starch, carbohydrates that are going to be broken down to power the growing embryo. There's an outer layer of this endosperm called the Alleron Layer, which you see right here. And that, as germination kicks off, that Alleron layer starts producing enzymes, particularly amylase, that is going to break down the starch. We then have a structure called the Scutellum, which is part of the cotyledon in our monocot here. And that Scutellum is going to help transfer the nutrients from the endosperm to the embryo. We then saw that there's a coleoptile, which is a sheath that is going to protect the growing shoot, which we call the plumule. We also have a coleorhiza, which is a sheath that is going to protect the growing root, which is called the radicle. And I should mention, I did start out by saying with germination, there are really three important things. And actually, let me write it here. To kickstart germination, right, we need the right temperature, and this may vary by species. We'll go a little more, I think we hit this later on when we talk about temperature. So the temperature is going to vary for what is the correct temperature or range of temperatures for germination for different species. We then need water. Water. And then we need air. Right, and that's oxygen. And for when we start photosynthesizing, we need some CO2. So we know those things right off the bat to start germination. And really the first step of germination is the seed imbibes water. So it takes in H2O. And by doing this, this is really going to kick off all these metabolic processes that are going to begin germination. So the first step is our seed takes in water. And then the first embryonic structure that emerges is, we'll draw it a little bigger, the radicle. And that's that primary root. And it grows through this coleorhiza. And then This radicle will continue to grow. We may start to see some secondary roots branch off. And next, the plumule begins to grow. That's the plumule, and that's the shoot, primary shoot. And that is growing. It's protected by a sheath called the coleoptile. And if we move right along, this gets bigger. We may see more roots, secondary roots. Eventually, this is going to break through the surface. And then we'll have the first true leaves that start developing. Right, so pretty simple pattern. The important thing is that these cotyledons stay below ground, and much of this germination takes place below ground. And it is just the plumule, that shoot, that emerges from the ground. Now we can contrast that with epigeal germination. So this is typical of our dicots, and this germination takes place above ground. Now our seeds are a little different in our dicots. We have two cotyledons, which take up the majority of the space in our seed. And there is some endosperm in these cotyledons, so they have some stored energy to help power the embryo. And then the embryo consists of primarily the radicle, just like with our monocot. There is a region of the seed stem below the cotyledons that we refer to as the hypocotyl. And then there's a region above the cotyledons that we call the epicotyl or the plumule, like you'll see over here in our monocots. So germination is a little bit different, at least the patterning. Again, we start, same thing, it takes in water, that first step. And what we see, just like in our monocots, the first thing that grows is our radicle. But really where this germination is different is, as this radicle is growing, instead of a plumule growing upwards, we have the growth or the elongation of the hypocotyl. So that would be the hypocotyl, and then attached to the hypocotyl are the cotyledons. So this is the hypocotyl. And these are the cotyledons. And this is kind of not the best drawing, but oftentimes this seed coat will come up with these still attached to these cotyledons, and these would be, if this is the seed here, these would be a lot bigger. Something like that more. But you get the idea. So what's happening is we have an expansion of this hypocotyl, and this hypocotyl expands, and eventually we get these two cotyledons that will emerge. And these guys are the seed leaves, like we talked about, and they will begin photosynthesizing in a lot of plants. Not all of them, but typically they will start photosynthesizing, and it's going to start producing food for the growing embryo, and this is continuing to grow, our radicle down here. And then from here, once this hypocotyl emerges, the epicotyl, or the plumule, will begin to grow, and then it's going to start producing true leaves. Right, and those true leaves will look different than our cotyledons. And it grows and produces more leaves. Right, so that is epigeal germination. Okay, so the cell is the basic structural and physiological unit of all living organisms. It is essentially the smallest unit of life. And when we are talking about cells, we can classify cells as either prokaryotic or eukaryotic. Now, prokaryotic cells lack organelles. And if you go all the way down to the bottom of this slide, you'll see that organelles are subunits of a cell that perform a specific function. You can think of them as little organs, right? They are these units, subunits of a cell that act just like you have organs in your body that have specific functions. You have organelles in a cell that have specific functions. And we're going to go over organelles throughout this lecture. So our prokaryotic cells, They lack organelles or other internal membrane-bound structures, and importantly, they have no nucleus. They are more primitive, so they're found in bacteria and blue-green algae. Now, our eukaryotic cells do have membrane-bound organelles and a nucleus, and they are our plant and animal cells, and it's the eukaryotic cells that we're going to be focusing on. Now, I say eukaryotic cells are both plant and animal cells, and we're in plant science. We're going to be looking at plant cells, so we should know the distinction between them, right? They have many things in common. They have a cell membrane. They have a lot of membrane-bound organelles that they share, but there are some distinctions between plant and animal cells. Importantly, plant cells have a cell wall, which we're going to learn about. They generally have a more of a fixed rectangular shape, whereas animal cells could be more amorphous, right? And that fixed shape is due to that cell wall. Plant cells have chloroplasts, which are the organelles where photosynthesis takes place. And we're going to learn about photosynthesis at the way end of this course. Animal cells do not have chloroplasts. They do not photosynthesize. Animals do not make their own food. Another distinction is that our plant cells have generally a large central vacuole. So that, if you look right up here, there's our large central vacuole. Animal cells will also have vacuoles, but they are not as big. Let's see if we can find one. There's a vacuole right there. So they're not a big central vacuole like our plant cell has. And there's a reason our plant cells have that large central vacuole. We're going to learn about it in a sec. So those are the important distinctions, right? Plant cells have a cell wall, animal cells don't. Plant cells have chloroplasts, animal cells don't. Plant cells have this large central vacuole, animal cells don't. Okay. So let's get in and talk a little bit more in depth with our plant cells. And really now we're going to be looking at the organelles of our plant cell. And we're going to kind of start working from the outside in. So as I said a couple times now, our plant cell has a cell wall. And this is at the outer layer of our cell. And the cell wall has a couple functions, right? First, it's going to protect the cell. So it provides some protection to that cell, and it provides an external structure that can lead to support of our cell. And in some of our tissues, which we're going to learn about in the next lecture, that external support is particularly important. And so there are additional structures built in, or chemicals built in to our cell wall that, I should say molecules rather than chemicals, that provide additional support. So it's protecting the cell and it's providing this external structure and support. It's our cell wall. It has two layers. We don't need to get really into that. We'll talk a little bit about it a little more when we look at tissues. But remember, cell wall, it's the outer layer, provides protection and structure. Now moving inward, we have what is referred to as the plasma membrane or the cell membrane, or it's also referred to as the plasmalemma. It's got three names. You just got to remember one. And so the cell membrane, or plasma membrane, is a lipid bilayer. So bilayer means two, lipids are fats, so it's just double layer that surrounds the cytoplasm. It's found interior to the cell wall. And the plasma membrane is a selectively permeable organelle. So it is essentially like the gatekeeper of the cell. It's going to regulate what can come in and out. That's what I mean when I say selectively permeable. It means it lets some things in to the cell and it blocks other things from coming into the cell. So it's helping to regulate what's going in and out of the cell in terms of proteins or nutrients, etc. Again, think of it like gatekeeper. Now, we will hear a term a little bit later called protoplast, and that essentially is the entire cell except the cell wall. So the protoplast is the plasma membrane, cell membrane, and everything interior, and it doesn't include that cell wall. Why that's important is later on when we're looking at tissues, there are certain tissues where the cells form and then the protoplast dies. So all you're left with is a cell wall. So when I get to that, when I talk about the protoplast dying, what I mean is essentially the entire cell except that cell wall. Okay, plasma membrane. Then we have the cytoplasm. So the cytoplasm is this thick fluid that is found inside the cell. It's composed of water and salt and some proteins. And it is essentially the fluid in which all the other organelles sit. Now, the cytoplasm has this movement called cytoplasmic streaming, and they don't entirely know how it works, but essentially that cytoplasm kind of moves throughout the cell, and in this way it's able to transport different nutrients and proteins and organelles around. You can see it down here, the streaming, and you can see these chloroplasts moving in the cell. That is due to this cytoplasmic streaming. But again, cytoplasm is the fluid in which all of our organelles sit. Okay. We also have in our plant cells what is called a Plasmodesmata. And this is a narrow thread of cytoplasm that is going to connect adjacent cells. And this is allowing our cells to transport stuff from cell to cell. Think of it as like a little tunnel, right? So you have your cells next to each other. Right here they have cell walls. There'll be an opening in the cell wall, which we call a pit. And running through that pit will be this plasmodesmata, this little thread of cytoplasm that is going to allow these cells to transport materials back and forth between each other. Again, it's like a little tunnel. All right, so the next couple of organelles that we're going to look at are all involved in gene expression and protein synthesis. So we'll get a little more in depth with that at another lecture, but I want to provide a little overview now because I think it'll be helpful to talk about it now, and then it's going to help you later on when we're learning a little bit more about it, and it should help you kind of remember these organelles. All right, so what we are talking about is gene expression. Man, I have bad handwriting. Gene expression. So we need to know a couple terms, and we have to a little background information. So your genes. A gene is a segment of DNA that codes for a protein. Right? It's kind of a molecular biological definition of a gene, but it's essentially what it is. Your genes, each gene is a segment of DNA that is going to code for a particular protein. And those proteins interact with each other, with the internal environment in the organism, the external environment, to create a trait, right? Whether it is leaf shape in a plant or eye color, height, etc. That is essentially what is happening when we're talking about genes and traits, right? Our genes are coding for proteins, and those proteins are interacting to create a trait. So our DNA is deoxyribonucleic acid. It is a molecule. And the important thing that I want you guys to know here is that our DNA is composed of this sugar phosphate backbone, and it has, let's see if I can draw it. That's not bad. So there are two strands of your DNA, right? And it has these two sugar phosphate backbones, and then between them are nucleic acid bases that join together. So if we zoomed in on this, we would have one backbone here, and then there are these nucleic acid bases that essentially join up together. Like so. And these bases consist of molecules adenine, oops, should actually spell it, adenine, thymine, guanine, and cytosine. ATGC. Now A always binds with T and G always binds with C, right? So you would have on one end here an A, one end here a T. So it would be a G, C, G, C like that, right? So you have your two strands of your DNA. They're connected by these nucleic acid bases. These bases always bind with certain ones. So A always binds with T, G always binds with C. Now, it is the segment, the pattern of these nucleic acid bases that creates a particular protein. And that's what our, it's essentially a code, right? So there are two processes. If we're talking about our going from a segment of DNA to a protein, there are two processes. The first process is transcription. And in transcription, we take DNA and we turn it into what's called messenger RNA. So we have a segment of DNA that is going to be turned, we're going to make a protein, right? So we have a segment of DNA, this section unravels, so here's our DNA here, yada, yada, yada. A section unravels, and we get one strand down here that has a particular code, right? So it could be A, G, G, T. And then what binds to it is messenger RNA. So RNA is another type of molecule found in cells. I need to look up one thing real quick because I don't remember it. That's it. Okay. So messenger RNA binds to our DNA. So our DNA strand unwinds, and then we have messenger RNA come in, and it binds to this DNA strand. And our messenger RNA is made up of these nucleic acid bases as well, right? So just like before, here's our messenger RNA coming in, and it binds here, and this is T. The difference, oh, actually, I'm wrong. Hold on. I am wrong with, oh, geez, that's a huge eraser. Let me change, oh, what's going on? Hold on. Let me see if I can adjust this so it's not extreme. Okay. All right. There we go. All right, let's get rid of this T. So a thing that I should make a note of here is that our messenger RNA, so we'll go real quick, mRNA has adenine. It does not have thymine. It has a different molecule called uracil. And then it also has guanine and cytosine. So essentially, you don't have thymine in messenger RNA. That thymine gets replaced with uracil. That's the difference, right? So you would have a U here. You have a C here. C here. And then you would have an A here. Okay, so you're going to make a protein. We take our segment of DNA, it unwinds. We have one strand of that DNA, a separate molecule called messenger RNA matches up with that strand of DNA, and now we have a segment of messenger RNA right here. And that is transcription. That is the first process of protein synthesis. And then the next process is this messenger RNA goes through something called translation. And that is where we take this messenger RNA and it essentially gets combined with something called transfer RNA. And through that process, we get a protein. And I actually have on the slide a little better, I can show you actually translation a little bit better than drawing it out here. But, so this is essentially just a quick little overview that I think will help when we're looking at these organelles, or at least help you remember them. Right? So they're all involved in gene expression and protein synthesis. And remember, a gene is a segment of DNA that's going to code for a particular protein, and those protein interacts to create a trait. Our genes are essentially, like I said, these segments of DNA, and DNA consists of two strands of this sugar phosphate backbone with these nucleic acid bases between them, and those bases form essentially a code that is going to allow us to create a protein, a specific code. The first process of this gene expression is something called transcription, and that is where our DNA gets turned into messenger RNA. So this DNA strand unwinds, and to one segment, a messenger RNA comes and binds to it. And though that sequence of these nucleic acid bases is the same for our messenger RNA, except that we replace thymine with uracil. So here's our DNA sequence, our messenger RNA sequence binds to it. That messenger RNA then goes through a process called translation, where we have a different type of RNA called transfer RNA that binds to the messenger RNA, and through that process it creates a protein. And we'll look a little bit more in depth at that. Okay, let's go back now. Okay, so that was a little bit of an aside, and like I said, you'll see that again. I kind of just wanted to give you a little primer on it. But I think it's helpful because it's going to, when I talk about these organelles, they're all involved in that process. So by understanding a little bit of that process, it should make the understanding of these organelles a little bit easier. So our nucleus is the command center of this cell. And in the nucleus, this is going to contain our DNA. Right? It has, and our DNA is combined and packaged into what we call chromosomes. But that in every cell, it has that nucleus, and that nucleus is containing that DNA. And it's going to control gene expression, that process that we just talked about, right? It also mediates the replication of DNA, right? When our cells are replicating, we also have to replicate DNA. So it's involved in that process of making new copies of DNA of the chromosomes. And when our cells split, so each cell has a copy. And it is also the site of transcription. So it's that first process where we're transcribing DNA into messenger RNA. So that is all happening in the nucleus. That's why we refer to it as the command center. There is also a section of the nucleus we call the nucleolus, and that produces ribosomes. And we're going to look at ribosomes right now. Right, so we had in the nucleus, if we're talking about gene expression, we had our segment of DNA that unwound. We had messenger RNA combined to it through that process of transcription. That messenger RNA then travels to a ribosome, and the ribosome is where translation occurs. Right? This is the actual site of protein synthesis. So how it works is that messenger RNA gets fed through the ribosome. And as it does, the transfer RNA comes and binds to it. Right? It binds, there's a transfer RNA, if you see here, has each transfer RNA has a different segment or code of these 3 nucleic acid bases we refer to as a codon. And as this sequence moves through, the messenger RNA moves through, a transfer RNA comes and matches up with that sequence. So here, a UUU, you would need an AAA transfer RNA, and it binds to it. And when it binds to it, on the other end of our transfer RNA is an amino acid. An amino acid is essentially a building block of a protein, right? Proteins are made up of chains of amino acids. So this messenger RNA is moving through. As it's moving through, our transfer RNA is coming and binding to a 3-segment nucleic acid section. When it does, it leaves behind an amino acid, and as we go through this process, we gradually build this amino acid chain, which is going to become our protein. So there you kind of see it in action, right? Our messenger RNA is moving through, transfer RNA is binding with the appropriate section, and it's leaving behind particular amino acids that are all going to combine together to make a protein. All right, so that's the ribosomes. Now, after our protein is made, there's a couple things that happen. So in the endoplasmic reticulum, and this is continuous with our nuclear membrane, so it's continuous with our nucleus. Our endoplasmic reticulum may have ribosomes embedded in it, it may not. But essentially, after our proteins are made, they get packaged. They get folded up and packaged by the endoplasmic reticulum, and then they are sent to the Golgi apparatus. So we have our proteins, they've been made by the ribosomes, they're now packaged up by the endoplasmic reticulum, sent to the Golgi apparatus. And the Golgi apparatus is the post office of the cell. So it receives the proteins, it's then going to modify them, sort them, and repackage them for either use in the cell or to be sent out of the cell to somewhere else. Post office. So all of those organelles I talked about, the nucleus, the ribosome, endoplasmic reticulum, Golgi apparatus, even nucleolus because it makes the ribosomes, are involved in that gene expression protein synthesis, right? Nucleus contains the DNA, it's where transcription occurs, so our DNA gets transcribed to messenger RNA. It moves to a ribosome where translation occurs, where our transfer RNA binds with our messenger RNA, creates a protein or a chain of amino acids. Within the endoplasmic reticulum, that protein gets folded up and sent to the Golgi apparatus. Our Golgi apparatus is then going to modify that protein, repackage it, sort it out, and then send it to either where it's going to be in the cell or somewhere out of the cell. Bam. All of those stuff. All those organelles. Okay. Moving on. We're going to talk about plastids. And probably the most important one that we're going to talk about is the chloroplast. So plastids are membrane-bound organelles that store products, or if we're talking about chloroplasts, are used in photosynthesis. An interesting fact about our plastids is that they likely were other organisms that existed freely. So what we think happened is way back, there was a eukaryotic organism, so an organism that had a nucleus, and it engulfed a cyanobacterium that it was otherwise would have eaten. But instead of eating it, it just adopted it into its body, and in this way, we got our plastids, right? So for chloroplasts, we had this eukaryote, it was going to eat this cyanobacterium, it engulfed it in its body, but instead of then breaking it down, it just incorporated into it, and that chloroplast then started producing food for this organism, and then it just lived on and got passed down through generations, and that's where chloroplasts derived. The same when later on when we talk about mitochondria, it's the same idea. We think a primitive cell engulfed a mitochondria or primitive mitochondria and then just incorporated into its body. Kind of a interesting evolutionary origin for these really, really important organelles, because when we're talking about our chloroplasts, this is the organelle that is responsible for photosynthesis. And photosynthesis is the process by which we get energy from the sun, and we use that to create food. Right? We get energy from the sun. The plants can then combine carbon dioxide and water to create glucose, which is a sugar molecule, which is the basis of food. It's really, really important. Kind of why they think this theory is, there's a number of interesting things about our plastids. One of the most interesting is that they have their own DNA separate from the nucleus. So besides chloroplasts, there are a couple other important plastids in our plant cells. There are chromoplasts, which synthesize and store pigments, which play a large role in our plants, as we'll learn. There are also leucoplasts, which just are storage plastids, and they can store starch or proteins, and they kind of have different names depending on what they're storing. But again, the most important is our chloroplasts, and that's at least what we're going to be focusing on. Now, I mentioned the mitochondria. The mitochondria is another organelle where we think it has that extracellular origin. So it was eaten by a primitive cell a long time ago and just incorporated into its body. And our mitochondria is the powerhouse of the cell. It is the site of cellular respiration. That is where we take our sugar and we break it down to create ATP, which is going to provide energy to our cells. It is how we are able to really do anything. All cellular activity is going to require some energy, and that energy is coming primarily from ATP, and it's through this process of cellular respiration. So again, it's a critical component of both plant and animal cells. Okay, we are coming to the end. Our last organelle that we need to be aware of is the vacuole. And again, in our plant cells, they have this large central vacuole that we don't see this large central vacuole in our animal cells. Our vacuole serves as a storage organ. So it can contain water or salts. It can even store toxic products that could damage the cell. So the vacuole is also important for maintaining structural support. So there's something called tonoplast, which is the membrane that surrounds this vacuole, and it controls the flow of water in and out of the vacuole. And it maintains turgor pressure. So turgor pressure is the force within the cell that pushes the cell membrane, or the plasma membrane, or plasmalemma, up against the cell wall. And through this turgor pressure, it helps maintain the rigidity of our plant. And if our plants lose turgor pressure, the cell wall the cell membrane can pull away from the cell wall, and that results in the wilting of our plants. Right? So this vacuole is really important for helping to maintain this turgor pressure and maintain the rigidity of our plants, particularly our herbaceous plants. Right? And if our plants don't have enough water, they don't have enough water to maintain this turgor pressure, and we see that in the plant starting to wilt. So that is it for our organelles. I got an extra YouTube video up here if you guys want to watch more. It is essentially just someone else talking about all these organelles. And that is that. So hopefully it's not too complicated. Again, you can kind of break it down into organelles that are responsible for protecting the cell, such as our cell wall, our cell membrane, and involved in some transport like the Plasmodesmata. Then there are those organelles that are involved in protein synthesis, gene expression, nucleus, ribosome, endoplasmic reticulum, Golgi Apparatus. Our plastids, such as our chloroplasts, are involved in photosynthesis. Mitochondria is the powerhouse of the cell. And then that large central vacuole is a storage organ, helps maintain turgor pressure, keeps our plants from wilting. That's it. All right. Next lecture, we're going to be looking at tissues.